Okay, could I have your attention, please? We'd like to get started again. So our next speaker is Josep Bassaganya-Riera, who is a professor at the Virginia Bioinformatics Institute and in the Department of Biomedical Sciences and Pathobiology here at Virginia Tech. He's already talked to you before. You've seen him. You've heard from some of his students. So you're somewhat familiar with him, but just briefly, he is the director of the Modeling Immunity to Enteric Pathogens, or MIEP, project. His background, he's got a DVM from the University of Barcelona. That really began our connection, I guess, as he brought Adri and Monica over. His PhD is in nutrition and immunology from Iowa State University. He's been at Virginia Tech a little over 10 years, rising through the ranks to his current position. Has a variety of research interests, including things like computational immunology, nutritional immunology. And so today, what he's going to be talking about is connecting data to health.
Thank you, Dave. And thanks, Madav, for setting the stage for my talk. As Madav was saying, this is really an integrated effort between several groups at the Virginia Bioinformatics Institute. It's very interesting. I find it very exciting that Kate joined us for this education activity, since she was very critical to get the project started. Now, it's interesting, we've prepared the presentations independently, and one of the slides that Madav used, I'm also going to use the first model that we developed with Kate, which, by the way, was not related to infectious disease, the main focus of modeling immunity to enteric pathogens. The focus at that time was inflammatory bowel disease. And what that shows us is, and I'll allude to that later, the portability and the way we are designing these models in a way that they don't become a one-show pony, that we can move from one infectious disease to another with perhaps some minor changes in the topology, with recalibration of data sets, and I'll show probably 2 use cases today. If we have time, I'll touch on the third one, as to how we have recalibrated models that you've seen throughout the week to answer different questions. Before we do that, I know that Dr. Allen spent a lot of time describing what the mucosal immune system is about. Of course, his focus was mainly on epithelial and myeloid cells, but I wanted to reiterate, because that's the basis of how we are constructing our models, at least our tissue level models. What are the key components of the mucosal immune system? And Adri alluded to those 2 compartments in his presentation, and you've seen those compartments embedded in the different SBML networks that we've shown throughout the week. We have the inductive sites where the immune responses are initiated, and in here you can see that we have the Peyer's patches as one of the inductive sites represented, the lamina propria as one of the effector sites. So the inductive sites is where the immune responses are initiated. The effector sites are where the immune responses take place. And so, for instance, in this representation, we have the colonic lamina propria, which is covered by a monolayer of epithelial cells. And Dr. Allen discussed the turnover of these epithelial cells and the importance of this turnover in the context of infection or just homeostasis. And under this epithelial cell monolayer, we have the immune cells, regulatory T cells, Th1, Th2, Th17. We saw the presentation from Dr. Youssef earlier this week, focusing on the beautiful work on Th17 differentiation. Obviously, that network that he reproduced would be embedded within this cell, which in turn is embedded within that colonic lamina propria or gastric lamina propria if we are exploring H. pylori infection. And what that shows is that there's an integration across the scales that is important from a temporal perspective, is important from a spatial perspective. And inductive sites and effector sites are connected. We have cells that are being activated, or where there's an antigen presentation in the Peyer's patches traveling to the mesenteric lymph nodes through the thoracic duct, accessing the bloodstream, and being then or migrating to the lamina propria to fight against viruses, bacteria, or to help maintain homeostasis. The mucosal immune system represents about 70% of the immune system in humans. It's a very significant part of the overall immune system. There's about 300 square meters of epithelium in our mucosal immune system. It's an impressive. We could pave several tennis courts with that epithelium, and we are replenishing that immune system on a daily or every 2-day basis. So those are some numbers, some food for thought. Of course, if we include the microbiota, where we have trillions of bacteria at the more distal stages of the mucosal immune system, it becomes an extremely complex system, where reductionist approaches can get only that far in comprehending function and comprehending phenotype. And so our laboratory has been working in the interface of genes, microbiome, diet, inflammation, and immunity over the last 12, 15 years. Our lab is called Nutritional Immunology and Molecular Medicine, and the nutritional immunology focus is represented by the interface between diet and inflammation. And just understanding that interface is extremely complex. And we soon came to the realization that if we were going to continue studying that relationship in a very specific way, slicing the problem so thin, then the problem was going to become meaningless. And I think it was in that context that we started to interact with NDSSL, with Kate and others to try to understand immune responses in a more systems level, not only at the gene expression level, but also at the cellular level and at the tissue level. And we started to build those more comprehensive computational and mathematical models of the mucosal immune system. And I wanted, before I get to the specifics of today's talk, because I think it's relevant to remember what this Modeling Mucosal Immunity Summer School and Symposium is about. We are at the last day of the summer school. Tomorrow we have the symposium ahead. In that context, we designed this education component to introduce immunologists to the latest methods and tools for using computational modeling. We wanted to present the MIEP and MIEP work to the wider audience, and wanted to disseminate those computational models of the gut mucosal immune system. And I hope we've made some progress in this regard. What have you learned so far? I hope that you have had a sense of what the mucosal immune responses are. I know the majority of you, by design, have an immunology training. I'm aware that there are some of you that have a more computational science background, and so we've tried to tailor this summer school towards the starting immunologists that may be interested in modeling, that may see value in the modeling approaches, that may see value in this integrated cycle of calibrating models, hypothesis generation, validating those predictions in the lab. And I hope that our models highlighted the architecture of the mucosal immune system. And as I was showing in my earlier slide, the inductive and the effector sides. If you think about the model that Adri presented of H. pylori infection, there were several compartments, and I'll show that picture again. There was the lumen, the epithelium, the lamina propria, and the gastric lymph node. So the gastric lymph nodes in that context would be an inductive site. The lamina propria would be an effector site. And so we've also talked about types of computational models of the mucosal immune system and tools. We've been using ODE models, we've been using agent-based models, we've integrated those models. We are making good progress. I hope that we'll have a multi-scale modeling paper as a proof of concept within the next month or so. And as Madhav said, there's a long way we need to go. That will be an initial proof of concept. We'll have to work very hard to get that platform and that model to the next level. We've also shown you how to build network models from data and theory. And I'll allude to that later. I agree with Madhav that you don't necessarily need data to build a model. In all our cases, we've used data, but what we haven't done is to forget about the theoretical framework. So we've integrated the theoretical components, the theory, with the data-driven components. And some of the models and use cases are more data-driven, some are more theoretical, but it is within the combination and the interface, and our tools take that into account, integrating data and theory to make predictions. And not to make predictions that are academic exercises. We are not in the business of running academic exercises, at least that's what I've been pushing the center. We are in the business of trying to solve problems related to human medicine. And so we hope that there's a translational value, and we hope that our models will help advance development of new therapeutics and vaccines. We've also alluded to mining immunological data sets using Cytobank or IPA. There was a talk about signaling regulatory networks and the models. And the tools that have been covered in some detail have been Copasi and EC Cell Designer. And within these, and particularly under Copasi, we've touched on calibration, sensitivity analysis, parameter estimation, simulation, model-driven hypothesis generation, and experimental validation. And what we are doing under MIEP is to build models that are portable and comply with standards, systems biology markup language for our OD models. The models of the immune system are applicable to infectious and immune-mediated diseases. And as I said, the first model that Kate constructed was actually an IBD model. It was designed to understand the underlying changes occurring in the context of inflammatory bowel disease. And he used data from a peak model of inflammatory bowel disease to calibrate that computational models. We hope that the models, or some aspects of them at least, can be recycled for new uses following recalibration with new data sets. And I'll show you an example as to how we've recycled the CD4 model that Adri presented on Monday, and we recalibrated that CD4 model with a data set specific to an IL-21 knockout project in the context of H. pylori infection to come up with a different or a new set of predictions that were then validated experimentally. And we are combining theoretical and data-driven approaches, and we are integrating data sets and exploring conflicting results in silico. This is the slide that Madhav presented and that I decided to present to show the legacy of how the story started. That was the first model of the lumen, lamina propria, and mesenteric lymph nodes, showing the interactions in the context of inflammatory bowel disease. And we hope to continue to develop this model further, not under this program, but probably through other programs. And that may seem out of context, but I have a question for you. I wanted to ask, what do you think there's in common in this context between the Chicago Daily Tribune and Google? And why would I even bring this up today? And anyone has a guess of what may have happened here? So, and the Chicago Tribune article states, Dewey defeats Truman. And I can say that it's a 1948 picture. Sorry?
You don't always get what's predicted. Is that what you're trying?
I think that's the point. In 1948, the Chicago Tribune predicted, based on inaccurate polls, that Truman was not going to win the election. Actually, Truman had very low levels of popularity after the war, and he didn't do very well. So there was an announcement, and they printed the newspaper saying Dewey was going to be the new president of the United States. And Truman was very happy to show that the next day when it was demonstrated that he in fact had won the election. Now, why is Google next to this picture?
Maybe their flu vaccine prediction?
Exactly. So Google made a big deal about their system to predict flu vaccine and flu infection outbreaks. This year, it has been confirmed that it was not so good. In fact, they were not able to predict the 2009 swan outbreak, even though this tool was already available, and their data doesn't seem to match very well the CDC. And so what we have here is that Google algorithms overestimated peak flu levels last year, and they didn't do a very good job in previous years. And I'm saying this, and I'm bringing this up even though we are not in the business under this program of studying flu outbreaks, because one of the reasons Google made a mistake was that they used the data, but they didn't consider the underlying theoretical framework. And so that would be an example in which data-driven approaches alone can result in big problems. So what we want to do is to build complementary strategies. Big data is great, and I think I'm not saying let's not use big data. I'm saying let's use big data not as a substitute for traditional data. Let's use that big data as a complement for traditional data. And there's a need for validating and integrating those data sets. And the modeling platforms that we are developing under MIEP provide an opportunity to integrate those theoretical frameworks as well as the data that is generated through flow cytometry, mass cytometry, maybe it's an ELISA data set, maybe it's a real-time PCR data set, or maybe they are histology pictures or clinical outcome results. We hope to be able to integrate those data sets with RNA-seq and high throughput kind of results. And we are doing this by integrating the knowledge from the literature, integrating the results from our own laboratory and other laboratories, and building those models, running the simulations. And eventually, and that was the first visualization, which I recognize it's not very good, of lesions formed in the gastric lamina-propria following H. pylori infection. Now we have better visualizations. And so I'll get to my use cases now. Adri alluded to the paradox of H. pylori. Is H. pylori a pathogen? Is H. pylori a beneficial organism? We know H. pylori is a bacterium, and there's no question about that. Another fact is H. pylori was classified as a type 1 carcinogen by the World Health Organization. And the approach that has been used to dealing with H. pylori based on NIH recommendations has been eradication. And in fact, many of the people in this room don't have H. pylori because the mechanism most doctors use when H. pylori is detected is let's get rid of it. Now, if we go to developing countries, the rates are going to be higher. Now, Martin Blazer at New York University has defined H. pylori as one of the most endangered species in our planet. And he has made the point that perhaps when we realize that H. pylori is needed, it might be too late. And the rationale for this is that there's an inverse correlation between H. pylori prevalence and the rate of overweight and obesity. And Adri alluded to a study that we conducted in mice showing that H. pylori colonization ameliorates glucose homeostasis through a PPAR gamma dependent mechanism. So H. pylori, particularly a type A negative strain of H. pylori, exerted beneficial effects. And so obviously H. pylori has some very detrimental effects linked to gastric cancer, lymphoma, and has some potential beneficial effects. And so what's going on? How do we understand this complex relationship between the host and the bacterium? And you saw this slide from Andrew's presentation. And he made the case that through his modeling, through his integrated modeling and experimental approaches, that perhaps treating H. pylori infection based on the results we have today might not be the best approach because H. pylori is only part of the problem. We may eliminate H. pylori, but H. pylori is inducing an immune response, Th1, Th17 response that may be involved in the lesion formation. And so can we modulate the host response underneath H. pylori infection rather than treating H. pylori itself, given that H. pylori is obviously exerting some beneficial effects? And the beneficial effects of H. pylori in the context of type 2 diabetes and obesity are linked at least to regulation of hormones produced by the stomach that regulate satiety. Ghrelin, leptin. And as a result, when H. pylori is not there, people tend to be less capable of regulating glucose homeostasis. They have to eat more. So, but in the context of H. pylori infections, we know that there's a significant influx of CD4 T cells. Cytokines and transcription factors activated in CD4 T cells are crucial to modulate myeloid cells, and there's a secondary myeloid cell influx. And so how can we target this immune response to H. pylori? And we believe a potential is to develop host-targeted therapeutic approaches. You also will remember this slide that was shown by Stephen and Adri. Those are simulation results of an EC, the agent-based modeling platform, showing that following H. pylori infection, we are inducing at about, after 30 days post-infection, we are inducing Th1, Th17, and there's a parallel induction of Treg cells. IL-21 is quite critical in the modulation of both Th17 and Th1 subsets. IL-21 helps in the maintenance of Th17 cells and impairs Treg homeostasis by IL-2 inhibition. IL-21, we've shown, is increased with H. pylori infection and correlates with level of gastritis, at least in the mouse model. So we had this network. Adri calibrated this network. He published this paper in PLOS Computational Biology. And the focus of that initial publication was the validation of a prediction between the plasticity of regulatory T cells and Th17 cells modulated by the transcription factor PPAR gamma. He used this network to then focus on IL-21, and he recalibrated the data set with IL-21 specific data to explore how the sensitivities change within the system following modulation of IL-21. And so this shows basically that we had calibrated the model initially for the publication of 2013. We recalibrated this model again with a data set from CD4 T cells extracted from H. pylori infected mice, and we had wild-type mice and IL-21 knockout mice. And what the data shows is, as expected, that IL-21 deletion causes, has the mice lacking IL-21 have lower levels of IL-17 and lower levels of interferon gamma when compared to the wild-type mice following infection with Helicobacter pylori. And so we use this data to recalibrate the model and to calibrate specifically the reactions that are related to IL-21. We run a sensitivity analysis to assess how sensitive are different molecules to the change in concentration of IL-21 following H. pylori infection. And we found that IL-21 activation is positively correlated with Th1 and Th17. So our in silico system was capable of regulating what was known, the theory. That was as a quality control, if you wish, of our modeling approach. And then we began to examine other transcription factors. We looked at STAT1, STAT3, ROR gamma C, TBX21, and our experimental data was matching the computational or simulation data. And what we found interesting was that IL-21 did not modulate FOXP3 expression during H. pylori infection. However, IL-21 had a significant impact on the IL-10 response by Th17 cells. In other words, our model predicted that IL-10 would be upregulated in the IL-21 knockout system, but there would be no differences in FOXP3. We then ran a follow-up experiment to validate this data, and FOXP3 was not changed, at least not statistically, is the graph on the left. On the right, we have a significant difference between wild-type and IL-21 knockout mice in terms of increased IL-10 production. So the deficiency of IL-21 in the context of H. pylori infection is inducing a regulatory, an anti-inflammatory cytokine. And this slide shows how close our experimental and predictive data was at the end of the experiment. And the question is, can we find a better, more targeted approach to reduce the inflammatory response triggered by H. pylori? Our data seems to suggest that IL-21 may be part of the story. This is H&E staining of stomachs from H. pylori infected mice. On the right, we have wild-type mice. On the left, we have IL-21 deficient mice. And as you can see, if you know how to read those histopathology slides, there's significant infiltration or leukocytic infiltration, a slightly enhanced mucosal thickness in the slide from wild-type mice, whereas a slide from IL-10 knockout mice, the mice lacking, IL-21 knockout mice, the mice lacking IL-21, and those are whole body knockouts, the leukocytic infiltration was significantly reduced, and the top figure shows the significant difference when comparing IL-21 negative and IL-21 positives. IL-21 is expressed at very low concentrations in the gastric mucosa following H. pylori infection, but it seems like even those very small concentrations of IL-21 have a major impact in modulating Th17 and interferon gamma and in the infiltration of leukocyte subsets. So we are now looking at molecule, and we've not made, we don't have the definitive answer yet, but actually Pfizer is developing a compound, a small molecule, for treating IBD. Obviously, IL-21 has been a target, a therapeutic target for inflammatory bowel disease, an immune-mediated disease. We are going to look into how this small molecule, what are the beneficial effects or neutral effects or negative effects of blocking IL-21 in the context of HP infection. If our model is correct, we should see that blocking IL-21 through this pharmacological approach eliminates lesion formation in the stomach. So this is a work in progress. I'm going to move now to the second use case, which also builds on previous presentations that happened earlier in the week. This is the model, the tissue level model of H. pylori infection. It represents the lumen on top, the epithelial barrier, the lamina propria, and the gastric lymph nodes, so the effector sites and the inductive sites. And you saw this slide from Adri showing the sensitivity analysis by using Kopasi approaches on the right, Anisi approaches on the left, indicating that at the initial stages of infection, the bacterium contributes to epithelial cell damage. At later stages of infection, when the infection becomes chronic, in the mouse model, it is the Th17 and Th1 subsets that contribute to that lesion formation, at least epithelial lesion formation. And in that model, we ask the question, what's going to happen if we delete PPAR gamma? And that actually is work that Kate initiated when she transitioned from the IBD model to the HP model. And I will not get into A, B, C, D, and F, but what I wanted you to take a look at is the prediction in terms of H. pylori loads in wild type versus myeloid-specific PPAR gamma null mice. Basically, what the model predicts is that the levels of H. pylori in the lumen will be greater at the chronic stages of infection than in tissue-specific PPAR gamma null mice. We've also looked at, if that's the case, is this effect mediated through the epithelial compartment, or is this mediated through a myeloid cell-related mechanism? So the epithelial compartment would be the top and is marked as 1 in subpanel B, and the myeloid-related mechanism is in the lamina propria and is marked as 2. And if you look at the results of the simulation, actually, if we infect with H. pylori wild type or myeloid-specific PPAR gamma null mice, we see that there's actually more in the more H. pylori. So that's different than the previous prediction. But if we infect and look at the M1 macrophage differentiation, we see that H. pylori has a pattern similar to what we had observed in the whole model. So we advanced experimentally, and we looked in mice that were uninfected. The left panel, the middle panel is infected wild-type mice, and the right panel is infected myeloid-specific PPAR gamma deficient mice. And what we are seeing is that the myeloid-specific PPAR gamma deficient mice have increased lesions, increased leukocytic infiltration, increased mucosal thickening in the stomach following H. pylori infection. And linked to the prediction that I was pointing out earlier, if we follow the bacterial loads, the amount of H. pylori in the stomachs of mice, and we followed this for over 6 months. We were killing subsets of mice serially. The prediction was holding true. The wild-type mice had higher loads. The PPAR gamma specific in myeloid cells had lower loads of H. pylori. And if we look at those macrophages, there's clearly H. pylori in the cytosol of the cell. And so we then run studies. The previous slide is in vivo. We run co-culture studies with macrophages from wild-type and tissue-specific PPAR gamma null mice. And the pattern was the same. The orange line represents H. pylori loads in wild-type mice co-cultured with H. pylori strain SS1. The lysozyme mice or tissue-specific or myeloid-specific PPAR gamma null mice is in blue. And so both at in vivo level as well as in co-culture studies, the deficiency of PPAR gamma in myeloid cells resulted in lower levels of H. pylori. And this was correlated with increased levels of iNOS in the tissue-specific PPAR gamma null mice. So we are still in the process of going back and forth with our model, tissue-level model, and our experimental validations. And we decided to use, and now you are probably familiar with several aspects of this pipeline. We decided to go to the big data. We decided to run some RNA-seq analysis to understand what was going on, why the deficiency of PPAR gamma in macrophages resulted in lower levels of H. pylori. What's the immunological mechanism underlying that phenomenon? So basically we ran a co-culture study, and then we collected RNA from those macrophages at different time points. And we ran this through the Galaxy pipeline. I know that you didn't do in-depth work with Galaxy, but you at least have been exposed. You know that there's about 50 local instances in the country. We have a local instance here that we use for our RNA-seq analysis. And this is available to you if you want to use. There are other very user-friendly graphic user interface-based systems to do RNA-seq analysis. But the analysis of that data was then done, at least the network inference component, through Ingenuity Pathway, and Kate did a good job yesterday presenting the capabilities of that tool. But the end result of the IPA analysis is a static network. We don't have the dynamics embedded. So we have the roads, but we don't have the traffic going through those roads. And so what we want to do, and I'll show you step by step the network that we built, was not only to create that static network, but to build a mathematical model from that static network so that we could assess the sensitivities of at least some of the key players that we believe may play a role in modulating responses to H. pylori macrophages. So one of the top networks that came up as regulated by responses to H. pylori was not actually an immune-related pathway, was a pathway related to metabolism. And so you may be familiar with the complex 1 through 5 oxidative phosphorylation proteins. They are embedded within the inner membrane of the mitochondria, and they are involved in producing energy. And what we found is that following H. pylori infection at different time points, there was an upregulation of genes linked to mitochondrial dysfunction and oxidative phosphorylation. We also examine other components of the network that have to do with innate immune responses and that may intersect metabolism-related pathways. We saw that some of the differentially expressed genes are the TLRs. Dr. Allen discussed TLRs, discussed NLRs. We had an NLR cluster, also differentially expressed, NLRP3 upregulated. NLRP3 was described yesterday as an inflammasome forming NLR, whereas NLRX1 was described as a regulatory or anti-inflammatory NLR. And what we see is the NF-kappa B pathway related genes are regulated, TRAF2, TRAF6, IRF7, STAT1 were differentially expressed, and then a series of cytokines, IL-1, TNF, IL-1, IL-6, IL-1 alpha and beta were upregulated. And this also was represented over time. We have 6 time points. I'm showing 2 time points here. And that information we are using then. So those are the static networks. And so here we have how things interact. We don't have the dynamics that underlie that interaction. So from this static network, we then want to build computational and mathematical model. And we are using ordinary differential equations and COPACI to build that computational and mathematical model to facilitate this going from the static network to the model. We've built a plugin that minimizes the work. Cassie Washington is working on this model. If we look at the fitting, our model was able to fit the experimental data well enough. There was a discussion about fitting, and there was a comment from Adri at about 5% difference from experimental and computationally generated data. And then we ran a sensitivity analysis in the context of this model. We looked at the local sensitivity for NLRX1, and what we are seeing that there's a relationship between NLRX1 and the kinds of molecules that would be involved in viral signaling cascades. Of course, H. pylori is not a virus, but nonetheless, the pathways that are being upregulated are viral signaling. So we would expect those pathways in the context of influenza infection, for instance. We also see NLRX1 and interferon gamma signaling. That there's an intimate relationship within our model. And the sensitivity suggests that there may be a role for NLRX1 in MHC class 1 signaling as well. So what the interpretation of this is that H. pylori has been viewed for decades as an extracellular bacterium. Now we have evidence that H. pylori can be found inside epithelial cells, can be found inside macrophages. And what this is beginning to point at, and it's a work in progress in its further development, is the potential for H. pylori to present not only through an MHC class 2 related mechanism, and all the CD4 work that we've done would allude to that CD4 related response, which would have been presented through MHC class 2, but also through an MHC class 1 related mechanism. And so if we look at NLRX1 expression and validation in macrophages, we see that when we compare wild type and PPAR gamma null macrophages, there's a difference in terms of NLRX1 expression. That's the validation by real-time PCR of RNA-seq data. And if we utilize NLRX1 knockout mice, we see that the deficiency of NLRX1 results in lower recovery of H. pylori from stomach following 3 weeks of infection. And now we are running longer studies because we are interested in the chronic stages of infection. And if we look at bone marrow-derived macrophages, the data represents well what we are seeing in vivo. So the modeling is pointing at new directions and new areas of exploration mechanistically in the context of mouse models. Now, I alluded to the MHC class 1 versus MHC class 2 presentation of H. pylori, and we've been trying to induce CD8 responses. So if there's presentation, if H. pylori is cytosolic, And if there is presentation through MHC class 1, we should see not only TH1 responses and TH17, we should see CD8 responses. We've had a hard time inducing CD8 responses, the kinds of responses you would expect in the context of influenza infection in the mouse model. So we've developed a pig model. Pigs are very similar anatomically, physiologically, immunologically to humans. And so we've developed in the context of this program a pig model of H. pylori infection. It's one of the first models developed. And this slide is showing how the gastric mucosa looks following H. pylori infection. As you can see here in this immunohistochemistry in panel A, H. pylori for the most part in the pig stomach, and it would be similar in the mouse stomach and the human stomach, is free floating in the mucus layer. Dr. Allen talked about the importance of the mucus layer as a means of protecting against infectious agents. And so most of the H. pylori can be found there. At the higher magnification, all these brown particles are H. pylori particles. Now, some of the H. pylori we find within myeloid cells. Inside, and this is in vivo extracted from pig stomach. So there's H. pylori inside myeloid cells. Those could be macrophages, could be dendritic cells. If we look at the immune responses to H. pylori following 49 days of infection, in pigs we're able to find an increase of CD8 responses at about 28, 35 days post-infection. And this increase in CD8 responses associated with H. pylori infection was correlated with increased expression of granzyme A, granzyme B, and perforin. Those would be the molecules linked to cytotoxic activity of these CD8 positive T cells. So we are starting to build a case for H. pylori not only being an extracellular bacterium, but also being intracellular and inducing the kinds of responses that one would expect in an intracellular context. And this again is an evolving story. So what will be the next steps for this use case? We've run some local sensitivities. We'll be running some global sensitivity analysis. I've shown you data for NLRX1. We'll run data for NLRP3 and LRC5, which by the way is linked to MHC class 1 presentation and was upregulated in that data set, not one. We'll generate additional in silico knockouts, and we'll integrate this gene expression model with the tissue level model by using the multi-scale modeling platform that Dr. Marate was alluding to earlier. And so that presentation, I hope, helps integrate the concept, the biological components of the program, at least the key biological components, with the multi-scale modeling discussion that will take place in the roundtable. We hope that the discussion is going to be very interactive, so we don't have a series of slides prepared for that talk. We hope that enough questions will be raised and enough issues and concerns, discussion of advantages of multi-scale modeling limitation will surface that will be able to add some value and hopefully synergistically think about how to improve the multi-scale modeling and take it to the next level. Really, the modeling immunity to enteric pathogens program, and I think this is applicable to all 4 programs under modeling immunity for biodefense, is a program that is at the cutting edge of experimentation, but each problem is a hard research problem. We are solving hard computational problems, we are solving hard mathematical problems, and we try to integrate them on a day-to-day basis. This is the acknowledgement slide for the MIEP team, but I wanted to also take the time, and I'll show this slide again in the symposium. Those people have been working behind the scenes very hard to make possible the education component of MIEP. And some people you've met, some people you may not have met, but I know they've been working very hard. Kim Barkowski, Adri, Dave Evan, Jim Walk. Kathy was involved actually in the preparation stages of this summer school. Rachel Robinson prepared some visuals, a nice video that was circulated. Tracy, Tiffany, Chris has been videotaping, Ivan has been taking pictures, and Josh has helped you with the IT aspect. So with that said, I'll be glad to entertain any questions. Thank you. Yes.
So that was great for the in the IL-21 knockout experiments. Well, one quick question: was IL-21 in the model before those experiments? Was it, or I know you said you recalibrated. Was it already in the model though?
It was part of the topology of the model, but the calibration data set that was used was not a data set that was really comparing wild type versus IL-21 knockout. So there may have been some IL-21 data in the wild type system,
Yeah.
but we didn't have a situation in which what happens when we take IL-21 out of the system.
Before you recalibrated it, was there any hint that an IL-21 knockout would have such an effect from in silico experimentation?
Well, the reason we decided to focus on IL-21 was because of the consistent, at least in mouse models, increase in Th1 and Th17. And so we wanted to go deeper as to what may have been modulating Th1 and Th17. IL-21 seemed an ideal target. We've been following the IBD story in parallel with the infectious diseases story. And we felt that even though it was expressed at very low concentrations, it had the potential to have a major impact. We also did some studies with IL-23, since it contributes to the maintenance of the Th17 phenotype. The results were not as striking. So we decided, and we generally, with the modeling, allows you to pursue several routes in a very inexpensive, cost-effective way. And so you can run several variations of the model, or even have some hypothetical reactions that you know is not in the literature, but what's going to happen if I connect this node to that node, how will the overall system change? With IL-23 it was not as successful, IL-21 was more successful, and therefore we decided to then invest experimentally to address further and to try to validate that prediction. Yes?
A question about the therapeutic approach that you're suggesting, that antibiotic treatment may not be the best approach because obviously it's the host response that in a chronic state maintains the inflammation in the gut. Are you suggesting that by blocking IL-21, you reduce the inflammation but then not to eliminate the gut itself? So would it be a combination treatment, or would it be just targeting the inflammation? Because if you still have the gut, a virulent strain of HB in the system, then
yeah, obviously we are simplifying the story.
Joseph, could you repeat that question?
Yeah, so the question is, are we proposing, based on the combined experimental and computational work, to address H. pylori infections only via host-targeted therapeutics and disregarding antimicrobial treatment, or are you considering a combination? And I think the question, the answer is it depends. The host pathogen interaction for H. pylori depends on how the host responds, and I think we've demonstrated that in a reproducible way. There's an effector response that contributes to tissue damage in similar way to what you would expect in a model of adoptive transfer of colitis, in other models that are considered more immune-mediated models. But then there's the bacterium. And there's a variety, a broad range of strains. And obviously, if you have a strain that you know for sure it's going to be pathogenic, it's been associated with cancer, you wouldn't want to just deal with the immune response. You would want to deal with the bacterium itself. But the reality, if you look at the epidemiology of H. pylori infection worldwide, not in this room a small proportion will be effective due to the treatment in the Western countries. But if you look worldwide, we have about 85% people infected. We have cancer developing or severe disease in about 10–15%. So what happens with the majority of population? Is that bug exerting beneficial effects? And if so, by eliminating it, perhaps we are making that population more susceptible to diabetes or to overweight and obesity. And so this is, I think, just the research at its infancy. But at some point, when there's enough data, and I would say that there's a need for some clinical testing, the paradigm of treatment may have to be reconsidered for certain strains of H. pylori. Yes?
So this actually also, when Adri was giving the talk about the H. pylori infection model, this also came up, and I was wondering what steps are you taking in order to incorporate more bacteria strains into such a modeling environment? And talk about not just the interaction of one bacteria, but several bacteria with the immune system and each other.
Yeah. So the question is, what steps are we taking to incorporate additional strains of H. pylori into our modeling and, I guess, experimental
Not so much H.
approaches?
pylori as other bacteria.
Other bacteria.
Other bacteria,
Yes.
other than H. pylori.
Yes, okay, so let me address the question that I thought you were asking, and then I'll address the question that you seem to be asking.
That's fine.
On the computational modeling, I'm going to say that we've not been working with one strain. We've been working with a European strain, 26695, SS1, J99, an African strain, and so we have a range of parameters that are tweaked depending on the strain. And if you go to the ANISI, the ANISI user interface was designed to be able to account for these strain differences. As to the second question, the gastric lamina propria and lumen is very different when compared to the colonic lamina propria. You will have trillions of bacteria, the colonic lamina propria. It's highly populated, perhaps 1,000 species. In the gastric mucosa, due to the conditions, the acidity, and the difficulty of most bacterial strains to survive in that environment, only a very small population of bacteria can be found. And so you may have about 200 species at the most, and H. pylori will be the dominant species. So the situation in terms of modeling microbial dynamics in the stomach is not as complex as modeling in the colon. Do we need to model? Possibly, yes. We have not done it, and that may be the next steps of our H. pylori modeling work.